Okay. Well, good afternoon, everybody, and either welcome or welcome back to the Harmful Algal Bloom Research Symposium. My name is Laura Essman, and I'm from the Indiana Water Resources Research Center at Purdue University. And I'm joined by Catherine DeLong from Iowa State University Extension and Outreach, and we will be your moderators for this afternoon session. The Algal Bloom Action Team is thrilled to be hosting this event for a second year, and we are glad that you were able to join us today. This year's symposium is organized into 4 sessions over 2 days. And if you joined us this morning's session was on HAB's monitoring and ecology. And this afternoon, we'll be talking about cyanotoxin treatment and detection this afternoon. Tomorrow's sessions will focus on forecasting and modeling HABs in the morning and emerging technologies for detection and monitoring in the afternoon. Each session will be facilitated by members of the Algal Bloom Action Team, which is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting this annual symposium, our team is developing a website of resources that can be used freely for outreach and education around harmful algal blooms in the North Central region. I encourage each of you to visit our website. Catherine's going to put that in the chat for us right now. And at the website, you can explore some of the resources we have to date, which include a What You Should Know fact sheet and a HABs Frequently Asked Questions database. And there are recordings from our bi-monthly webinar series, as well as last year's virtual HABs Research Symposium. Before we get started, just a few housekeeping items. We will have 4 30-minute presentations this afternoon with a 15-minute break between the second and third talks. At the conclusion of the 4th talk, we will hold a 30-minute presenter Q&A panel. Pay attention to this part because it's important. To have your questions addressed during the Q&A panel, please post them using the Q&A panel function in your Zoom window as we move through all of these talks. It's perfect and it worked out great this morning if you include your affiliation with your question as well as which speaker the question is directed towards. If there's a question already posted in the Q&A panel and you have the same or similar question, all you have to do is use the Zoom upvote function on the question to help ensure it gets answered because the most popular questions move up higher in the panel. Please note, the Q&A panel function is separate from the chat function in Zoom. If you have technical difficulties, please contact us through the chat function and we will assist you as best we can. Once we have completed the presenter Q&A panel, we will have some closing comments from Rebecca Power, Director of the North Central Region Water Network. All of our symposium presentations are being recorded, and we will post them on the Algal Bloom Action Team website at the conclusion of this event. As a reminder, this afternoon's presentations focus on cyanotoxin treatment and detection, and I will now turn things over to Catherine to introduce our first speaker.
Thanks, Laura. All right, if we can go to Hamed's slide. Perfect. Okay, great. Our first speaker this afternoon will be Hamed Haidari. Hamed is a PhD candidate at the University of Nevada, Las Vegas. His previous works were on biodegradation of EDCs or endocrine disrupting chemicals such as phenol and biphenyl A. He recently submitted a peer-reviewed paper concerning the absorption of Gen X. Haidari's dissertation focuses on photocatalytic degradation of cyanotoxins that released during these HAB incidents. So I will now turn it over to Hamed. Hamed, you can start sharing your screen when you're ready and start presenting.
Thank you. Okay, you guys can see my screen?
Yes, it looks good.
Perfect. Hello, my name is Hamid Haydari. I'm working with Dr. Ikala Khan at University of Nevada, Las Vegas to do catalytic degradation of cyanotoxins by bismuth tungstate. First, I'm going to talk about the cyanotoxins that I selected for this study and explain why I selected them. Then I will name some of the prevalent treatment methods used for cyanotoxin mitigation. I will introduce perovskite and give you a little background on it, and I will share some of the results I got using bismuth tungstate to degrade cyanotoxins. Then I will discuss the path forward, and at the end, you can see the references I used to make this PowerPoint. Cyanotoxins. Cylindrospermopsin is a hepatotoxin containing a hydroxyl methyl uracil moiety connected to a tricyclic alkaloid moiety. Anatoxin A is a bicyclic secondary amine alkaloid. And Microsystem LR is a hepatotoxin and has a cyclic hepatopeptides with 7 distinct amino acids in its structure. The rationale behind selecting these 3 cyanotoxins is the enone functional group in all of these cyanotoxins in their chemical structure. The hypothesis is that photocatalytic degradation of these 3 cyanotoxins starts with the enone functional group. In 2017, the United States Geological Survey Agency conducted a survey of 11 major coastal and inland rivers of the United States, and cyanotoxins were detected in most of them. Circles classifies the cyanotoxins synthesis gene detection, and the triangle in the circle represents the prevalent cyanotoxin that was detected. As you can see, the microsystem and anatoxin are prevalent in most of inland and coastal waters. Warmer waters, exposure to sunlight, excessive amount of nutrients leads to growth of cyanobacterial species that release cyanotoxins. Cyanobacteria produce elevated levels of toxins under a stressful environment. So if we have a stressor, it will act as a facilitator for cyanobacteria to release more toxins. Cyanotoxins are categorized in 3 categories: dermatoxins, hepatotoxins, and neurotoxins. And some of them are cytotoxins, which means that they target several organs. Human exposure to cyanotoxins are from ingestion, inhalation, and skin contact. Common symptoms for dermal contact includes skin irritation, rash, eye inflammation, and lacrimation. Now, I'm going to talk about Cylindrospermopsin. Several cyanobacteria species produce Cylindrospermopsin, including Dolichospermum, Cylindrospermum, Pantothrix. The molar mass of Cylindrospermopsin is 165 gram per mole, and it is highly soluble in water. And there are 5 known congeners of Cylindrospermopsin that is cited in the literature. For anatoxin, Cylindrospermopsis, Afaminoxenon, Omezakia are the prevalent cyanobacterial species that produce it. The molar mass of a toxin is 415 gram per mole. It is highly soluble in water. It is known as very fast death factor. Why is that? Because it killed mice within 2 to 5 minutes. There's no regulatory guidelines for toxin, Silentospermopsin, and MCLR inside the US, but there are with the US EPA, but Ohio Department of Health set the advisory level of 20 microgram per liter for toxin. Approximately, there are 14 cyanobacterial species capable of producing cyanotoxins. You can see some of them listed here. Microsystem LR is the most studied cyanotoxins among all. The molar mass for MCLR is 995 gram per mole. It is highly soluble in water, more than 1 gram per liter. It's bioaccumulative, and the main target for MCLR in a human body is liver. It also have adverse health effects on kidney and reproductive systems. The drinking water threshold that the World Health Organization set is 1 microgram per liter for MCLR. As you can see here, here's the structure of MCLR and 6 microsystem congeners. As you can see in the figure, there are 6 groups that are influential in naming microsystems. They are shown with the arrows. For instance, the difference between MCLR and MCLA is the Z group, where MCLR has an arginine, while MCLA has an alanine in their chemical structure. Membrane filtration has been used to remove cyanotoxins, but the downside of using membrane filtration is that the cyanotoxins remain a problem in brine. Also, biofilm formation reduces the flux rate, and it is cost intensive and energy intensive. Different types of adsorptions, carbonaceous adsorbents, such as granular activated carbon, powdered activated carbon, and agricultural-based activated carbon, graphene, as well as iron-modified adsorbents were used to remove cyanotoxins. The removal efficiency of granular activated carbon and powder activated carbon was not promising, and their performance decreased in the presence of competing ions. In addition, adsorption only transfers contaminant from one medium to another, so the problem remains. Photolysis can degrade cyanotoxins to some extent, but the process time is long. So complete abatement of cyanotoxins has been attained by photocatalytic treatment in minutes. That's why I chose photocatalysis for degradation of cyanotoxins, because it will convert cyanotoxins into less or non-hazardous compounds, byproducts. Titanium oxide is the most studied photocatalyst for cyanotoxin degradation. It has an energy band gap of 3.2 electron volt, which is in ultraviolet spectrum. Ultraviolet irradiation occurs only in 4% of solar light spectrum, and it is cost intensive to provide required energy with ultraviolet lamps. Titanium oxides usually dope with other metals such as silver to benefit from the light absorption capacities of them to capture sunlight irradiation to be in solar light spectrum. Thus, we decided to use perovskite-like compounds that are strong in visible light absorption and revolutionized solar panels. The perovskite structure has the general formula of ABX subscript 3. This structure accommodates most of metallic ions with significant anions in periodic table. The metal substituted for A sites acts as extrinsic donors, and metal substitute for B site act as extrinsic acceptors. The simplest member of Variability family and most common perovskite is bismuth tungstate. You can see here a structure of perovskite and an example of perovskite mineral. Bismuth tungstate has attracted extensive attention due to its excellent intrinsic physical and chemical properties such as ferroelectric piezoelectricity, catalytic behavior, and nonlinear dielectric susceptibility. In addition, it has the highest photocatalytic activity among bismuth-based oxides under visible light irradiation. It has a low band gap of 2.8 electron volt, which is in the visible light spectrum. It has degraded organic contaminants such as phenol, methyl orange, and in minutes over 90%, and it achieved over 90% degradation. Here you can see some structures of bismuth tungstate, plate structure, particle structure, flower-like structure, and nanocage structure. In my work, my experiment, I use particle structure as my starting point. And here you can see the Cyanotoxin MOPS degradation by particle-sized particle structure bismuth tungstate. In order to make sure there's no adsorption happening, I had a sample running in the dark to make sure that there's no adsorption. The initial concentration of cyanotoxins were 15 nanogram per ml, and it was a mixture of all three cyanotoxins. So I didn't do them separately. The dosage of bismuth tungstate was 1 molar. The experiment lasted 4 hours, and as you can see, only 20% of Cyanotoxin MOPS was degraded after 4 hours. As you can see, the concentration of cyanotoxins did not change in the dark, indicating adsorption is not happening. The slight changes in is probably due to instrumental and analytical precision errors. Here you can see the degradation of toxin efficiency by bismuth tungstate. As you can see, bismuth tungstate was performing better for toxin, and it got 25% removal using the same conditions. The bismuth tungstate dosage was 1 molar, and with the initial concentration of 50 nanogram per ml for anotoxin. And again, the sample showed that there is no adsorption happening in the dark. And here, sorry, after 4 hours, the Microsystem LR was only degraded 15%. It wasn't promising for Microsystem LR under the same conditions as I stated before. The situation with bismuth tungstate is that the overall that I got from it is that it's not as promising. This particle size structure is not as promising as we were thinking. So moving forward, we will investigate various dosages of bismuth tungstate to make sure that if we increase the concentration of bismuth tungstate, the dosage of bismuth tungstate, it will it will improve the cyanotoxin degradation or not. Other structures of bismuth tungstate will be explored, and we will especially nanocage superstructure, and we will explore a dopant for bismuth tungstate to improve cyanotoxin degradation, to see if it can improve cyanotoxin degradation efficiency or not. And here are the references that I used. Thank you for listening.
Thank you, Hamed. I know Anne and I both just reminded everybody in the chat box to please submit your questions in the Q&A function. And for those of you who are technologically challenged like myself, the upvote is the thumbs up. So just click on that. Let me see if I can successfully share my screen again. Hang on here.
Laura, we did have one question that maybe because we've got lots of time, would it be okay if Hamed answered it?
Sure.
It's from Scott from KDHE. He asked, can you explain congeners and its significance to your presentation, Hamed?
Okay. So congeners is like the same chemical compound, but it has differences in functional groups on the chemical structure. And since Microsystem LR is the most prevalent one that was detected in coastal and inland waters across the United States, our focus was on Microsystem LR, but since they share similar molar masses, probably whatever works for Microsystem LR can work on other congeners. However, because they have different functional groups, the pKa for them might be slightly different. So in that case, in that perspective, congeners matter.
Thank you.
Thank you, Hamed.
Thank you.
I would like to go ahead, and I know we're getting a little bit ahead of schedule, but I don't think that matters since we don't have a concurrent session. So I would like to introduce our second speaker, Nafisa Khan, who is a master's student in the Department of Civil, Environmental, and Infrastructure Engineering at Southern Illinois University, Carbondale. She is a graduate research assistant in Dr. Ja Leo's lab. Her research focuses on mitigating harmful algal bloom, for example, inactivating HAB causing cyanobacteria, degrading cyanotoxin and removing nutrient phosphorus from water by magnetic nanoparticles in photocatalysis process. Prior to joining SIUC, she received her Bachelor of Science degree in Civil Engineering from Bangladesh University of Engineering and Technology. Today, Nafisa is speaking on efficient harmful algal bloom mitigation by magnetic visible light-driven photocatalysts. And Nafisa, would you like to share your screen?
Yes, sure. Hello, everyone. My name is Nafisa Khan. I'm from Southern Illinois University, Carbondale. And today my topic of the presentation is efficient harmful algal bloom mitigation by magnetic visible light-driven photocatalysis. Yeah, I am having an issue. The slide is not moving forward. Let's see. Yeah, I'm having an issue, so let me just see what happened.
Sure, Nafisa, would you want to try one more time? If not, I have your presentation open and could advance for you.
Yeah, I think it's working now. Yeah, sorry for the interruption. So the introduction. So in fresh water.
Can you please share your screen again?
Oh, okay, so sorry. Yeah. So in bigger screen, it's not, I don't know why the slide is not changing. So is it okay if I put it like this?
I can go ahead and share it for you here. So one moment and I will.
Actually, there's a bit modification from the one I sent. That's why.
Are you working on two monitors, Nafisa?
Mostly my one is mitigation part. Yeah, it's working now. Sorry. Yeah, it's working now. Yeah. So in fresh water, harmful algal bloom or HAB are associated with high concentration of cyanobacteria. This is actually or most commonly called the blue-green algae. So in late of summer, we see this green slimy layer mostly in some parts of the lakes. So it's actually overgrowth of algae. So the question is why harmful algal bloom or HAB occurs? For one of the reason is excess nutrient like phosphorus and nitrogen coming from wastewater, agriculture runoff cause this proliferation of cyanobacteria. So these can be quite harmful because cyanobacteria can produce cyanotoxins like microsystem, cylindrospermopsin, and that affects liver, digestive system, respiratory system, and thus harmful to human and also to ecosystem. So, and also it adversely impacts the recreational activities in the US. The objective of my study is to mitigate this issue, harmful algal bloom, by magnetic photocatalysts. Here I'm using gamma iron oxide, titanium oxide nanoparticle. So the mitigation process here it would be including inactivating the toxigenic cyanobacteria, photocatalytic degradation of cyanotoxin, absorbing the nutrient phosphorus by the nanoparticles. Another aspect of this study is to recycle the nanoparticles by magnetic separation because this nanocomposite has this iron oxide which has a magnetic property. So our aim also is to recycle the nanoparticles after water treatment, and it would reduce the addition of extra solids into the lake water. So this slide is actually a schematic diagram of mitigation mechanism. So under sunlight, the nanoparticles may produce reactive oxygen species that would damage the cyanobacterial cell wall or can also directly physically attack the cyanobacterial cell, absorb the nutrient phosphate from the lake water. And this reactive oxygen species can under sunlight would degrade the cylindrospermopsin and microsystem cyanotoxin that is produced by cyanobacteria. So the question is, I've mentioned that I'm using this gamma iron oxide titanium oxide nanoparticle. The question is why I'm using this specific nanoparticle. So titanium oxide is mostly investigated as a photocatalyst for inactivating cyanobacteria. However, titanium oxide, it works on this UV range, and UV range is actually a certain fraction of the solar range. So the nanocomposite gamma iron oxide titanium oxide would lower the band gap, and it would also work in the visible light region. And as the gamma iron oxide is coupled with titanium oxide, as mentioned before, it would have this magnetic property, and through magnetic separation, it can be recycled and reduce the extra solid and reduce the toxicity to lake ecosystem. And also another thing I would like to mention that it has been that iron oxide and titanium oxide is biocompatible and have low toxicity. So it would be good for ecosystem. Now to the next slide. So here, as I've mentioned before, in the material, I would be using this gamma iron oxide titanium oxide nanoparticle. So in the lab, it has been synthesized through precipitation and calcination process from ferrous chloride, ferric chloride, and titanium chloride. So this is a figure in the left side. This is a figure of the fabricated nanoparticle. And the right side is the transmission electron microscopy image, TEM. The size of the nanoparticles were measured as 15 to 100 nanometer. So this nanoparticle was characterized by this TEM, energy dispersive X-ray spectroscopy, EDS, and dynamic light scattering, DLS test. So in the right side, this is the result is from the DLS test result, where percent mass is actually plotted against diameter. However, from the graph, we see that only 8.45% mass percentage of the nanoparticle had diameter below 100 nanometer. It could be due to the storage issue and the aggregation of the nanoparticles. From EDS result, the weight ratio of titanium and iron is calculated as 1:1 in the produced nanomaterial. So now comes the water sampling locations. So for my study, I have collected water samples from 2 lakes, Carbondale Reservoir and SIUC Campus Lake, Southern Illinois University Campus Lake. So both lakes are actually used for hiking and natural trip purposes. And SIUC Campus Lake have had HAB outbreaks over this like late summer and early fall. And so these the water samples from the campus lake was also used for inactivation to see how nanoparticles work with the lake water in a batch experiment. So this is the right side. This is the figure of this is an image of the campus lake over this period where there was outbreak of algae and the water samples collected. So from July to October, the water quality parameters that were measured was chlorophyll A, phosphorus concentration, and cyanotoxin concentration. Chlorophyll A concentration, the highest one was calculated as, experimentally we got 1.8 milligram per liter, and phosphorus concentration was got 2.6 milligram per liter. Both values were actually more than the eutrophication level of lake water, and the lake was already in eutrophic zone at that time period. And also cyanotoxin concentration, microsystem, from July to September 2021, it was from 9.95 and to 38.8 ppb. And for this high level of cyanotoxin, the lake was actually closed over this time period for recreational purposes. And its cyanotoxin concentration was more than recreational threshold advisory of 8 ppb. And also another cyanotoxin, saxitoxin, it had around 0.03 to 0.17 ppb, but its level was lower than the recreational advisory threshold, that is 0.8 ppb. And water temperature during that July to September was time period, the highest water temperature was around 31 degrees centigrade. Now next to the another lake, from July to October, the chlorophyll concentration and phosphorus concentration was determined, and its value was lower compared to the campus lake. And in this lake, there was no overgrowth of algae was actually observed. However, its chlorophyll A concentration and phosphorus concentration was more than the eutrophication level. And for my study, apart from using water sample from the lake, now cyanobacterial pure culture was also used for inactivation of the cyanobacteria within the nanoparticle. The two cyanobacterial culture, Microsystem aeruginosa and Cylindrospermopsis rasiborski, was actually purchased, and to see how the nanoparticles inactivate samples that only have one specific cyanobacteria. So under microscope, the Microsystem aeruginosa had spherical cells, and the Cylindrospermopsis rasiborski, it had a straight filamentous type of cell. And also another thing of using this cyanobacteria cell culture was to see that how the size of how the shape of the cells actually is affected by the nanoparticle, whether it differs or not. It was also another reason of using this two different cyanobacteria pure culture. So this is the method portion. Here, water samples were interacted with the nanoparticle, put in the vial, and was placed on a tube revolver and was kept under visible light or simulated solar light. Cyanobacteria was quantified by quantitative polymerase chain reaction, Qpcr, and DNA was extracted. For this, DNA was extracted by Qiagen DNase power water kit. For Qpcr, cyanotoxin genes were utilized for targeting cyanobacteria. 16S rRNA gene is actually for all cyanobacteria species, accounting for both toxigenic and non-toxigenic species. MCYE or NDAF gene is actually for microsystem and nodular ring producing cyanobacteria. CYRA gene is for Cylindrospermopsin producing cyanobacteria, and RPOC gene was a species-specific gene, and it was only used for Cylindrospermopsis rasiborski, the cyanobacteria pure culture. And SXTA gene was for sexitoxin producing cyanobacteria. So from the Qpcr, amplification curves were obtained, and CQ value was also obtained from the Qpcr results. And for cyanotoxin degradation, for cyanotoxin concentration measurement, it was quantified by liquid chromatography with diode array detection as shown in this image in our lab, and phosphorus concentration was measured by HAB spectrophotometer. So now comes the result and discussion part. The pure cyanobacteria pure culture, Microsystem aeruginosa, was interacted with the nanoparticle concentration of 100 milligram per liter for 1 hour for both visible light and under dark condition. And the cell, and before the interaction with our nanoparticle, the cells were counted, and also after the interaction with the nanoparticle, the living cells were counted using hemocytometer under optical microscope. And from here, from this table, it is observed that under visible light condition, there was a reduction of average 21.8% cell reduction. And in the dark condition, there was also some cell reduction around 11.5%. However, visible light, under visible light, the cell reduction was more. And this is the Qpcr result. And Qpcr result, we have this before interaction without nanoparticle value, and after interaction with nanoparticle value. So before interaction, the CQ value is around 11.9, and after interaction is 26.7. So actually lower CQ means that higher concentration of cyanobacteria and vice versa. So it's actually consistent that before interaction, the cyanobacteria concentration was more, and so the CQ value is lower. And from that, the gene copies of M. aeruginosa was calculated, and from that, the reduction in gene copies were expressed in percent inactivation, and it is seen that the percent inactivation under visible light is around like 5 log or 99.995%. Similarly, for this another cyanobacteria cell culture, C. rasiborski, similar concentration of nanoparticle 100 milligram was used under visible light, and cell counts were calculated, and the average percentage cell reduction under visible light was around 33%. However, when the cyanobacteria pure culture was interacted with the nanoparticle, but in dark condition, there was actually no visible reduction in the cell counts, no significant reduction in the cell counts under dark condition. And this is the Qpcr result. So for Qpcr result, same 100 milligram per liter nanoparticle was used for 1 hour under visible light. And without nanoparticle, we have this CQ value of 7.14. And after interaction with nanoparticle, it has higher CQ value, 35.8. And it also similarly, gene copies were also calculated, and average percentage inactivation was also close to 100%, that is 5 log. It was also 5 log reduction. And now this part is the relation between chlorophyll A concentration and gene copies. So we know that cyanobacteria contain chlorophyll A, and so higher concentration of cyanobacteria would also have higher amount of chlorophyll A. And this relation was plotted to see how chlorophyll A is actually connected to gene copy. So for the first graph, that is for 16S rRNA, that accounts for all cyanobacterial species, both toxic and non-toxic toxigenic cyanobacteria, the R-square value was around 0.6. And however, for MCYE or NDAF, the R-square value was quite low, 0.3. That means that the second graph is not a good fit, that chlorophyll A and gene copy is not that good correlated. And similarly, for CYRA and SXTA gene, we also see very low R-square value. Similar conclusion can be made that chlorophyll A and the gene copy for these genes are not a good fit. And this is the result of cyanobacterial inactivation from the campus lake that was collected. So for this Qpcr result, both dark condition and visible light condition was considered, and 2 different concentration of the nanoparticles, 1 gram per liter and 100 milligram per liter were taken to see how the change of concentration of the nanoparticle changes in the CQ value, or there's change in the inactivation, percent inactivation. So from this table, it is seen that from MCYE for MCY or NDF gene under dark condition for 1 gram per liter, the percent inactivation is around 87%. However, for 100 milligram per liter, it's quite high, around 99.86. And similarly, for visible light, the percent inactivation is almost same for 1 gram per liter and 100 milligram per liter nanoparticle concentration. So it is seen that for dark condition, the 1 gram per liter inactivation efficiency is actually less compared to 100 milligram per liter nanoparticle concentration. And now for this SXTA gene, similarly, those 2 concentrations of the nanoparticle and dark and visible light condition were considered. And we see that from this table that for 1 gram per liter, the percent inactivation is quite low compared to 100 milligram per liter nanoparticle concentration. It's around 46%. And for 100 milligram per liter, the percent inactivation was around 98.5%. And under visible light for 1 gram per liter and 100 milligram per liter, the inactivation percentage was quite similar. So now next slide is about the representation of the CQ value and all those genes, unrelated to the 16S RNA, MCYE, NDF, and SXTA. So from here, it is seen that after the interaction for all of the genes, the CQ value was higher compared to the before interaction value. However, for MCYE or NDF, we see higher CQ value, very close to 30. And so it could be related to that 16S rRNA actually accounts for all the cyanobacteria species, both toxic, toxigenic, and non-toxigenic. So MCY or NDAF gene containing cyanobacteria are usually smaller in shape, like microsystem is spherical in shape. So this shape also has, when they are inactivated, when they're interacted with the nanoparticles, the inactivation efficiency would be more if they have smaller surface area. And so that's why it's quite high. And similarly for SXT, we also see higher inactivation. However, for 16S rRNA, we see lower value of CQ compared to the other two genes, because 16S rRNA actually have like all those cyanobacteria species that, and also can also have this that are not account for all of this, which may have different shapes. So that's why there's this difference in the values. And now comes the photocatalytic degradation of the cyanotoxin. MCLR was purchased, and a 200 microgram per liter concentration was made. And similarly, with the same nanoparticle concentration, 100 milligram per liter was used for 1 hour. And we see that with nanoparticle, the degradation was around 68.7%. And however, without nanoparticle, we also see some degradation. It was 48.6. However, it was lower without the nanoparticle. And as I have mentioned before, that phosphorus, excess phosphorus, is one of the reason of overgrowth of algae and also the toxigenic cyanobacteria. So it's important to remove this additional or excess phosphorus that's causing this HAB. And so for these 3 types of water samples were used. First one was in DI water, only phosphorus was added, and initial concentration was 200 microgram per liter. And when it was interacted with the nanoparticle, the average percentage phosphorus removal was 34.1%. And for the Southern Illinois University Campus Lake, the phosphorus removal was around 19.6%. And from Carbondale Reservoir, the average phosphorus removal was 42.7%. So actually, in SIC Campus Lake, as I've shown in the image, during that HAB outbreak, there was its color was very green compared to Carbondale Reservoir or the phosphorus added in DI water. So when I am interacting the nanoparticle with the campus lake for phosphorus removal, in the same time, the nanoparticles is inactivating the cyanobacteria and inactivating and absorbing phosphorus. So it could be one of the reasons that the phosphorus removal for campus lake is lower compared to the other 2 samples. And as I have mentioned previously, that one of the aspects of using this nanoparticle is it is magnetically separable. So here, this is in this figure, there is in the left side, there is a nanoparticle suspension in DI water, and a magnetic tube has been put beside the vial, and the figure shows that all the nanoparticles have accumulated on the side. And this is how these nanoparticles can be recycled and regenerated and can be reused. So, and now the conclusion. So in conclusion, Qpcr method can be used for targeting toxic cyanobacteria, but as I have shown in the figure, that chlorophyll A alone cannot be used to predict HABs. And lab-made, this nanoparticle has been used to inactivate Microsystem, Nodularin, Cylindrospermopsin, and Cyanotoxin producing cyanobacteria species, photocatalytic degradation of cyanotoxin, MCLR, and adsorb nutrient phosphorus. Better inactivation efficiency under visible light has been observed, and it indicates that this technology can also be applied under sunlight for HAB mitigation from surface water. And this HAB mitigation process can be used to protect public health in recreational activities in surface water. So these are the references I have used for my study. And that's, thank you. Thank you all.
Thank you, Nafisa. I think we have time for one question before we go to break. And it is from Dominique from Kuka College. She asks, how did you determine if the cells were living using microscopy? And someone else added, did you do live dead counts?
I actually did mostly did live count, because when the nanoparticles were interacted with the cells, for example, microsystems had this spherical shape. Some when I did after interaction, when I have seen under the microscope, some shapes were deformed, and some were not in their regular shape, and excluding those, I only counted other cells.
Okay. All right. Well, thank you. So
you.
this concludes the first half of our afternoon session. As a reminder, we'll answer the majority of the questions after all four speakers have presented. So you are going to have a nice long break. Our second to last speaker of the day will start promptly at 2:25 Central. So we
Catherine,
look forward to seeing you all back then. Yes.
I think we should just go with a 15-minute break and get started early.
Oh, okay.
So if we want to make that even, we can come back at, is it 1:55?
Sure.
Okay. We'll
Okay,
see
well-
everybody back in 16 minutes.
All right. All right, welcome back, everyone. Our next speaker is Jin Lee. He is a research scientist and adjunct assistant professor in the Department of Agricultural and Biosystems Engineering here at Iowa State University. His research works to improve our understanding of the link between microbiomes and their surrounding environments by developing advanced laboratory and computational methods and by systemic analyses of biological and environmental big data obtained from agroecosystems and aquatic ecosystems. So welcome, Jin. You can go ahead and start sharing your screen and begin. That looks good.
Thank you. Do I sound okay?
Yep, sound good.
Okay. Hi, my name is Jin Lee, and I'm working as a research scientist under the supervision of Dr. Adina Hao here at Iowa State University. I'm grateful to have a chance to talk at this symposium, and actually I'm learning a lot thanks to the wonderful presentations. Here today, I'm going to talk about forecasting cyanoHABs in Iowa lakes using a random forest model. Through this presentation, I would like to share what we learned from our 3-year project funded by EPA. So in the summer, lake beaches are popular places for recreation in Iowa. I think it will be the same everywhere. There are 39 sites regularly monitored by the Iowa Department of Natural Resources. The Iowa DNR takes samples once a week between Memorial Day to Labor Day, and they measure microsystems and water quality, some water quality parameters. When the microsystem level exceeds the EPA 2019 guideline, which is 8 micrograms per liter, they issue an advisory and close the site. Overall, 23 advisories were issued in 2021 and 12 advisories in 2020. I believe this type of process is quite similar to other states. So when we think about the cyanoHABs, the first question people may ask can be, do cyanoHABs only occur in a few certain lakes? So this figure shows the frequency of the events that exceeded 8 micrograms per liter microsystems in each lake and each year since 2006. Although 8 micrograms per liter is a quite recent guideline, and as far as I know, previously it had been 20 micrograms per liter, but we counted the number of events exceeding 8 micrograms per liter. So as you can see, some lakes always had a problem, while some lakes never had a problem. In the case of Black Hawk Lake here, it had frequent cyanoHABs occurrence until 2016, but we could not see any problems after 2018. And here the Pine Lake here, it rarely had problems, but somehow it had severe problems near 2016. So the answer to the question would be no. It happened to any lake and is quite unpredictable. The following question might be, are those cyanoHABs occurring, are those cyanoHABs occurring lakes located in a certain region in our state? Again, the answer would be no. The red circles in this figure indicate the lakes that had cyanoHABs events between 2018 to 2020. So it can occur throughout the state. Then what does trigger cyanoHABs? There have been a number of research papers that studied these elements. The bottom line so far, as far as I understand, cyanoHABs occurrence is a highly complicated process involving lots of factors. First of all, there should be biological factors because microsystems are produced by some genera of cyanobacteria, such as microcystis, Planktothrix, and so on. Those biological factors are affected by chemical factors. Nitrogen, phosphorus are well-known parameters affecting cyanoHABs occurrence. And maybe land use nearby the lake can be a factor impacting chemical profile in the lake. Also, climate, including precipitations and temperature, can be influential factors. Can we predict cyanoHABs if we measure as many components as possible? That was our question, and we aimed to build a machine learning-based prediction model for cyanoHABs occurrence. Most of all, it would require as many samples as possible from dozens of lakes, then as many parameters as possible should be measured. After establishing the data set, machine learning algorithms can be applied to the data set to extract useful information and to build a prediction model to aid decision making. I believe 4 key elements need to be well prepared to successfully apply machine learning algorithms to environmental data sets. Those are proper algorithms, sufficient data, high quality data, and a proper question. Please let me go through these elements one by one. So for the selection of algorithms, there was not much thing to consider because there are already all advanced and fancy algorithms there. So as far as I know, artificial neural networks tend to outperform other algorithms for unstructured data, such as images and text. But decision tree-based algorithms are considered the best for structured and tabular data. So this chart shows the evolution of tree-based algorithms over the years. The title of this presentation was saying we were going to build a random forest model, but after comparisons and our trials, we chose to use the XGBoost algorithm. So the title should be changed. XGBoost model is an advanced type of decision tree model, and it was introduced in 2016, and it is the most advanced one at this moment. So we chose XGBoost algorithm. So we have checked what would be the proper algorithm. So now let's move to the quantity part. So all our work could have been done thanks to the collaboration with the Iowa DNR. So every year, the Iowa DNR takes samples once a week on every Tuesday and Wednesday from 36 lakes, starting from Memorial Day to Labor Day, which is about 15 to 16 weeks. The number of samples was over 500 each year, and we could analyze about 1,500 samples in total for 3-4 years. Of course, analyzing more samples might be possible and better, but that was the most number of samples we could handle. So after having enough samples, what we needed to care about was the quality of data. So first, the sampling part was totally relied on the Iowa DNR. They had a very well documented procedure and already had over 10 years of experience. So, for sampling, each person was in charge of one of 4 routes and took 4 or 5 samples per day on Tuesdays and Wednesdays. Right after taking samples, the samples were transported to the merging point. At our end, picking up the samples at the merging point, subsampling and filtering was done on the same day. And filtered samples were stored in a deep freezer for molecular biological analysis later. Chemical analysis were also done as soon as possible. Microsystems were measured by the Iowa DNR, and other chemical analysis were conducted in a lab at Iowa State University. For every week, there were biological duplicates and technical duplicates for quality control. Also, we had 4 gene targets to quantify. Bacterial 6' rRNA genes were the target to measure the total number of bacterial cells in each sample. The other 3 targets were the MCYA genes originated from different genera, which is Microsystem and Planktodrix and Anabaena-like. The MCYA genes are one of the genes involving in microsystem production. Because we had multiple targets and over 1,000 samples to run, a high throughput Qpcr was used for gene quantification. So we could be confident that our data was consistent and reproducible. The remaining element was having a proper question. So when we imagine the best model, the ideal model, the model would be something that can predict the actual concentrations of microsystems. I believe that can be possible someday if an enormous quantity of data is available. But unfortunately, as you can see in this graph, the microsystem concentrations in over 1,000 samples were close to zero. Saying that it was unfortunate may be inappropriate because it is great news for the people in Iowa. But this imbalance was the biggest challenge to building a model. What we could do instead was sorting the samples into two groups, the safe group and the hazardous group, based on the EPA guideline. Maybe that can be the most straightforward information to aid decision-making, whether it will be safe or hazardous. Another aspect when we think of the proper question is how to couple input parameters with the target class. We had two classes, safe and hazardous. So as you can see here, if we couple the input parameters of week 1 to the class of week 1, it may not be a prediction model. So considering all the samples had a 1-week interval between samplings, we coupled the input parameters of week 1 to the class of week 2. So in this way, this model can predict whether the microsystem level will be safe or hazardous in the following week. So it was possible because all our samples had the same interval between samplings. Finally, all elements were ready to go after setting the question as, can we predict whether the microsystem level would be safe or hazardous in the following week by using the input parameters. So after all the measurements were collected and organized in a tabular form, more relevant or important parameters were selected through the feature selection process. Using the selected parameters, the model was trained. Before moving to the result, I have to say that we used all 2018 to 2020 data for training because the number of hazardous cases was too small. But soon we are going to add 2021 data as the test set. So please excuse that the result I'm going to present is only for the training set. To consider both aspects of non-linearity and linearity, we used a random forest and lasso regression for feature selection. The overall importance in the bottom, considering both aspects, was calculated. As the result, microcystis MCYA gene copies, pH, the ratio between MCYA genes and 16S RNA genes, chloride, TKN, and the herbaceous area were chosen as input parameters. Please note that the feature importance here does not mean the selected parameters are always more important than other parameters. These selected parameters were chosen because those can explain the given data set efficiently. So it does not apply to, it does not generally apply to other samples. So it was quite interesting to see the statistical differences in selected parameters between the safe and the hazardous group. The mean pH and MCYA gene copies, the ratio between MCYA genes and 16S RNA genes, and TKN were significantly higher in the hazardous group, while the mean chloride concentration was higher in the safe group. So since the result of the XGBoost model can just be shown as performance metrics, as just numbers, let me show a single decision tree as an example. This is not the final model, but it is the simplest and easiest way to understand the decision tree-based models. So when a set of measurement is given to the model, the first criterion was the ratio between MCYA genes and 16S RNA genes. This is about how much portion in the bacterial community was composed of toxic cyanobacteria. If the ratio is less than 1.26%, the next criterion was TKN. If TKN concentration was less than 2.6 milligram per liter, almost all cases were classified as safe in the following week. When TKN was over 2.6 milligram per liter, the following criterion was chloride. If the chloride concentration is less than 6.53 milligram per liter, more cases were classified as hazardous. In the other branch, all cases were classified as safe in the following week. So this is how a decision tree is made and work. So here are the performance metrics of models. For comparison, two other types of models, logistic regression and ARIMA, were also tested. The classification result can be filled in the table shown below. The accuracy is the overall performance, which shows how much the model predicts safe as safe and hazardous as hazardous. It means 91% of cases were correctly predicted. The specificity means how much model predicted hazardous as hazardous. The number 0.73 indicates that 73% of hazardous cases were correctly predicted, but it also means 27% of hazardous cases were predicted as safe. The lower performance of the hazardous class was expected because the number of hazardous cases was so small. If there are more hazardous cases available, the performance can be improved. The ROC AUC is another performance metric that we can see the overall performance easily. If we randomly guess whether it will be safe or hazard next week, the chance would be 50%. And the area under the curve, AUC, is 0.5. If the AUC value is 1, that indicates the model is 100% perfect. So the AUC value close to 1 can be considered as a better model. So the AUC value of our XGBoost model was 0.91, which outperformed other models. As wrapping up, the next two slides are a kind of my personal thought or epilogue. In 2015, I began to interested in applying machine learning algorithms to environmental data. So as the result, in collaboration with two environmental consulting companies, my very first machine learning model paper was published in 2016. So when I searched the literature to write the paper, I noticed that there had been not many trials that applied machine learning to environmental data. But I believe you remember that 2016 was the year of AlphaGo. So before the match against one of the best Go players in 2016, most people thought it would need 10 more years for computers to solve such a complex problem. But surprisingly, AlphaGo won 4 out of 5 games against a professional player. Now, we can easily search those type of papers. I could easily find recent papers that applied machine learning for the HABs problem in like 1 or 2 minutes. So once a model is designed, its performance can be evaluated, and it can be helpful for humans' decision. And humans can find an error in the algorithm, which can improve the algorithm. Better designed algorithms lead to better performance. Better performances help humans make better decisions. Humans who make better decisions can find the situations where algorithms fail. Those failures are again helping to improve the algorithm. I believe that's what exactly happened to AlphaGo. In the fourth game, after the professional player made the move 78, so-called the divine move, AlphaGo acted weird and lost the game. But that failure helped AlphaGo improve more and more. So I hope our current model would not be the end of our trials, even if there are many challenges ahead, such as the imbalance in microsystem concentrations. And the other unexpected thing was we didn't see the climate parameters in the selected features. So because we could easily think that the precipitations easily affect the HABs occurrence. So there are many things to improve, but I hope this type of trials can be continued. So in summary, selected parameters through feature selection showed significant differences between a week later safe and a week later hazardous groups. And the machine learning based data mining can be a promising tool for forecasting cyano HABs and environmental decision makings. And finally, it requires to build a systematic and comprehensive databases based on long-term monitoring to figure out and resolve those challenges in cyano HABs prediction. That is all I have today. Thank you for your attention.
Thank you, Jae-Jin. Just like the rest of the presenters, we'll give you one question before going on to our last presenter. The one for you that has the most votes is from Bob Clement with the EPA. He asks, are we looking in the wrong places? beaches are open and quite different than the many small bays in a lake, should we be looking at the worst case bays to accelerate our knowledge?
If I get the if I understand the question correctly, the problem is if we can know when and where it would happen, then we can find the worst cases and to improve our knowledge faster, if that's the case. But the thing is, it's not possible to know where would be the worst cases, right?
Mm-hmm. And I think too, he's asking about specifically, are there, you know, where you're taking the sample from specifically in the lake? Could that be a factor in what you're seeing?
Yes, so that could be the factor. That's true. As I mentioned in the slides, so the Iowa DNR has a well-documented procedure. So they have there's a kind of procedure how much deep they go into the lake. I think there's a certain distance mentioned in the procedure, which I don't remember. And yeah, they have very specific rules, and they try to take the samples at the same point as much as they can. So yeah, mostly like 10 to 20 meters from the shoreline, I guess.
Okay, great, thank you.
Jin, I'll ask you to stop sharing your screen and we will move on to our next presenter. Thank you very much.
Thank you.
Okay. I would like to introduce our final speaker for the day, Brian Haggard. Brian has degrees in life sciences, agronomy, and biosystems engineering, and he is currently the director of the Arkansas Water Resources Center within the University of Arkansas System Division of Agriculture. And he is a professor in the Biological and Agricultural Engineering Department within the College of Engineering at the University of Arkansas. Brian considers himself a biological engineer with a twist of ecologist, hydrologist, and water quality specialist focused on how what we do in the landscape influences water quality, nutrient transport, and trends over time. Recently, he has shifted towards a focus on harmful algal blooms and is trying to understand what triggers toxin productions in lakes. So today, Brian is going to give us a talk titled Microsystem Thresholds and Bioassay Response at a Recreational Impoundment Lake Fayetteville. And let's see, let me stop sharing and I will allow Brian to take over.
Thank you. Let me get this one going. Are we good there?
It looks great.
Okay, fantastic. Thank you very much for the introduction. Yes, appreciate being the last talk today. And Jin's talk was really interesting to me because it focused on, you know, lakes across a whole state. And I'm going to narrow down focus and kind of present some, you know, decision tree models very much like he did, but something specific to one lake. And so as you see there, the title of my presentation is Microsystem Thresholds and Bioassay Response at a Recreational Impoundment Lake Fayetteville. Lake Fayetteville is a small lake. It's roughly about a kilometer square in surface area. The catchment area is only 24 kilometers. It's 50% pasture, 39% urban, and only 8% forest. So it's, it's an impacted reservoir. Previous studies on this lake have shown that about 50% of the nitrogen inputs are retained within the lake and actually lost through denitrification, whereas 90% of the phosphorus that comes into the lake, accumulates in the bottom of the lake. And this city is owned by the city of Fayetteville. It was originally their water supply back in the, intended in the 1950s, 60s, because it was constructed in 1949. However, in the late 1960s, Northwest Arkansas as a whole switched its water supply to Beaver Lake. And so back in 2018, I bought an Abraxas plate reader with the intent to start measuring microsystem using the ELIJA kits. And as we were getting used to those methods and everything, I had a freshman engineering team that needed an honors project. And I told them, I said, well, let's just, let's go out to local lakes. In particular, I want you to pick Lake Fayetteville, collect water samples, bring them back, and we'll analyze those water samples for microsystem. I told them, don't have high expectations of finding anything over the reporting limits or even over the method detection limits of that particular method. And so they went out and sampled in December of 2018, and we found about a 0.5 microgram per liter of microsystem in Lake Fayetteville. Okay, that's not high, but we didn't see microsystem during the winter time in any of the other lakes. And so that we just kind of committed that to memory. And then in 2019, kind of once, later in early March, we went and took our first sample at this lake. And then in April, we took another sample. And then about in the May timeframe, we started sampling this lake on a weekly basis. And we've been sampling this lake on a weekly basis since then. And so what we've seen is we've seen peaks in microsystem during the growing season, peaks each year that we've monitored, but each year those peaks are different. And in 2020, we started collecting samples specific to the surface scum. And really that's where we're finding microsystem in this lake is, we have several occurrences of greater than 50 micrograms per liter of microsystem in the surface scum. And that data is not shown. And we really haven't tried to pair that data with any of the other data we're collecting at the lake. So I'm going to focus really on the data that's shown here, which is right below the surface. And when you kind of zoom in to this data, microsystem shows an early summer and a fall peak in 2019. This is definitely prevalent in 2020. And if I had the data from this fall, which I know what the data tells me, this graph wasn't updated for this presentation. We saw again in late November another little peak in microsystem, though not the same magnitude of an increase that we saw in the 2 previous years. This was really kind of an interesting observation, and it changed how long we ended up doing our nutrient bioassays this past summer. Our adaptive plan, and I say adaptive because we have, things keep continually change as we learn more information about the cyanoHABs at this lake. But our adaptive plan to study in cyanoHABs at Lake Fayetteville was the first thing we did is we needed to understand sampling and analytical variability to help us with future project planning experiments in our statistical comparisons. The next thing we did was we focused in on data from the 2020 growing season to understand what physicochemical properties and algal parameters showed thresholds and hierarchical structure with microsystem. And then I say hot off the press because my graduate students sent me this analysis yesterday, and I dropped it into this presentation, is looking at a kind of a separate subset, looking at the data on a calendar year basis. We wanted to understand how these thresholds and hierarchical structure between microsystem and all the parameters we were measuring varied between years. That is, looking at each year independently, 2019, 2020, and 2021. And then what did these models look like when we pulled all the data together? And then finally, this past year, we did a series of bioassays to try and understand how nutrients, nitrogen and phosphorus influence algal, cyanobacterial growth, and microsystem concentrations in production. And in the future, we're interested in looking to explore various treatment or management options, to mitigate the microsystem that we're seeing at Lake Fayetteville. In 2019, we went out and sampled one day at 11 a.m. and, we ran microsystem, and it ended up being 1 microgram per liter. And that day I talked to Alan Wilson down at Auburn University, and I was telling him about Lake Fayetteville, and he's like, hey, Brian, go grab a sample and overnight it to me. I'd like to look at it. And so we went back later that afternoon around 4 o'clock, grabbed another sample to ship down to him in a cooler. And I told my crew, I'm like, well, if we're sampling for Alan, let's go ahead and collect another sample and analyze it for us too. And when we did, one of the things we noticed was, at 11 o'clock, microsystem was 1 microgram per liter. And then at 4 o'clock that same afternoon, it was 11 micrograms per liter. And so that kind of got me thinking, it's like, is this diurnal variability or is it just sampling variability? And so we decided to look at sampling and analytical variability, just to try to make sure we're doing things right. So what we did was what you see here when you kind of look at these stats is the first box plot is going to be we're calling that between bottles. So basically we reached down into the lake water and we collected 15 different water samples. The second one is within a bottle, and there we just randomly selected one of the 1 liter bottles that we collected from the lake, and we poured back 15 different subsamples for microsystem analysis from that bottle. And at this point in time, we were following the methodology that was provided to us by Abraxas, and we're just freestalling around 2 mils of water. And so we poured, that's 15 different 2 mil samples poured back from 1 bottle. And then we randomly, the third box plot that you see there is we randomly just picked a vial and we measured, microsystem from that 1 vial 15 times. And again, a lot of y'all are probably way more experienced with microsystem analysis than I was at this time, but we just wanted to get a handle on the sources of variability, in the studies that we're going to be doing and really understand that. And so the one thing I would say is the good news is we feel really good and confident about the analysis. We don't think that there's a lot of analytical variability of what you're seeing in those vials after you put it through 3 freeze-thaw cycles. But when you look at the other 2, the between bottles and the within bottles, we found that there is a lot of variability in the microsystem concentrations that we're observing, both when we collect 15 independent samples from the lake, and from each of those samples pour back 2 mils for the freeze-thaw cycles and then analysis, or if we do 15 different samples from within a bottle. You can see there, we actually were fortunate enough to kind of sample over 3 different ranges of microsystem. And no matter what range you're looking at, you can see an order of magnitude difference in the concentrations that we were measuring. And so what this got us thinking was, and maybe we should have just reached out to a group like this 3 years ago or 2 years ago when we were doing this, is so then we did a little study and we said, well, what if we pulled a large lake volume from Lake Fayetteville, and then we churned it and mixed it or homogenized it really well. And then we poured back numerous 2 milliliter sample volumes, some 5, some 10, 20 milliliters, 60 mils, and even 300 milliliters. And we put all those through the freeze-thaw cycle. And we were trying to see, it's like, is there an optimal, subsampling volume from our lake samples that we need to be putting through the freeze-thaw cycle? And, we kind of centered on somewhere between 20 to 30 milliliters at a minimum is what's needed. And so now when we're doing this analysis and putting samples through the freeze-thaw, is we're using 60 mil glass vials. We fill them up about halfway, so they have about 25 to 30 mils of water in them. And that's what we put through the freeze-thaw cycle and then turn around and do the analysis. Like I said, hindsight, after we did this, I talked to Alan, and Alan's like, yeah, we've always been, pouring back, 20 to 30 mils, and that's what we've been saving and putting through the freeze-thaw. And so maybe other labs are following this same process. And if you are, that's great news. If you're using a volume greater than 20, but at least for us, this in 2020 kind of changed the way we were collecting our data from our lab experiments and our environmental monitoring. So the next thing that we did at Lake Fayetteville was we zoomed in on this data set right here. Is I just kind of clipped the all the water chemistry and physical chemical properties and algal properties from any samples collected from March of 2020 through September 2020. And we used CART analysis, that's classification and regression tree analysis, to look at thresholds and possible hierarchical structure with microsystem using temperature, nutrients, and algae as our dependent variables here. And we pruned our trees really well, I would say. What I'm going to show you is just the top 3 splits that we had in the data. We used a minimum bucket size, or we required a minimum of 5 samples to the left or right of any split for it to be significant. And it had to increase the complexity parameter by at least 0.1 in this case on what I'm going to show you right now. And all the models that I'm going to show you explained over half of the variation in microsystem data in our observations. And so the first one is our strongest independent threshold was with temperature. And so when we use temperature and then we use the nutrient chemistry and algal pigments or raw fluorescence, this was the model that we saw is, when water temperature was greater than 27 degrees Celsius, that's when we had high microsystem concentrations for the most part. You pull that data to the right and it plots up with the raw fluorescence of chlorophyll A. And this is measured using one of those handheld cyanofluors. We found that to be a really interesting and useful tool for us in our lab. And so when you look at the raw fluorescence, and then this data split at roughly 3,500 raw fluorescence units, and you take all the samples greater than that and you move them over to the right, and the data splits again with the particulate nitrogen to phosphorus ratio, and it splits at a ratio of about 59. And to the left of that, you have an average concentration of around 3 micrograms per liter, I think 3.6 micrograms per liter, and to the right of that, you have an average concentration around 1.9 micrograms per liter for microsystem. And so this is how kind of how our model built up. If you lowered the required increase in the complexity parameter, the tree would get more complex. And so what these models look like really varies based upon how you're selecting your minimum bucket size as well as your change in complexity parameter. And we're going through right now trying to statistically optimize that to kind of make sure we're picking the best pruned tree, so to speak. If we dropped out water temperature from the 2020 database and just looked at nutrient and algal pigment raw fluorescence, this was the model that we observed. The first step in the model was microsystem with nitrate plus nitrite nitrogen concentrations. And so when nitrate plus nitrite nitrogen was less than 0.07 milligrams per liter, that was when we typically had the highest microsystem concentrations. And so, at some level, that's kind of contrary to what a lot of people would think. It's so like when nutrients were the least in this reservoir, that's when we're seeing microsystem pop up in measurable concentrations. We take that data when the nitrate concentrations were less than 0.07 milligrams per liter, and the data actually split there with the ratio of phycocyanin to chlorophyll using the RFUs from the cyanofluor. And so here, basically what we saw was when the ratio was less than 1.5, we had increased microsystem concentrations. You can see here in this box. When it was greater than 1.5, we tended to have microsystem concentrations less than 0.5 micrograms per liter, with the exception of 4 data points up here. But when we take all of this data when we take all the samples that had a phycocyanin to chlorophyll A RFU ratio of less than 1.5, the data then split again with just looking at the chlorophyll A RFUs. And, and this split in the data was around 4,500 RFUs. And so when you had low nitrate plus nitrogen, a ratio less than 1.5, but your chlorophyll RFUs were less than 4,500, you had an average microsystem concentration of less than 1.1 microgram per liter. It was when you had more chlorophyll or a greater amount of algae growing or cyanobacteria growing in the lake, that's when you ended up with the higher microsystem, when you had these low nutrients and a phycocyanin to chlorophyll ratio of less than 0.1. This was one of the ones we thought was most interesting and possibly potentially useful from a lake management standpoint. If all we did was use the raw fluorescence data of phycocyanin and chlorophyll and that ratio, we observed these thresholds in this structure here. And so basically, if we had high chlorophyll RFUs exceeding, roughly 4,200 RFUs, that data went over to the right. That tended to be when we saw the greatest microsystem concentrations. And then when you look at that data, you see 2 different splits with the phycocyanin to chlorophyll ratio. And so basically, when that ratio is less than 0.6, you don't tend to have high concentrations of microsystem. But when that ratio is greater than 1.5, you don't tend to have high microsystem concentrations at Lake Fayetteville either. It's when that ratio is between 0.6 and 1.5 that you had the greatest microsystem concentrations in this lake. And so we thought that was really interesting because, the cyanofluor could be a really good tool, for forecasting on hypereutrophic reservoirs like this, where you have, a lot of human interaction and you're constantly having to give warnings about potential harmful algal blooms, at least to have kind of an idea if your phycocyanin to chlorophyll ratio, if you got this data is really easy to get at, it might be a way for the lake managers to kind of, know when to issue advisories and when not to have full advisories. And so we thought that was interesting. And but if we looked at the cart output for all data, and again, this is where, this is my graduate student, Alyssa Ferry. She's getting her master's in crop, soil, and environmental sciences. She was using all the data from each of those calendar years, as well as, looking at each individual year together, and dropped that into cart and kind of kicked out the output. I mean, when we do all of the data together, this is hundreds of data points that we have going into this model. At this point, we have 3 sites that we sampled, plus the 3 years of data. This model is explaining around 25% of the variation. And you can see that, the first driver was dissolved inorganic nitrogen when it was greater than 0.5 milligrams per liter. We had a mean concentration of microsystem around 0.2 micrograms per liter, and that represented about 43% of the data. But when concentrations were not greater than 0.5 micrograms per liter, that is, when concentrations were less than DIN concentrations were less than 0.5 milligrams per liter. We ended up with these splits over here, and you can see, 2021, we put some, some more categorical variables in this analysis as well. And when we looked at all the data, 2021 popped out as being completely different from the other two databases, which really didn't surprise us based upon what we saw. And but you see this model here, if we look at 2019 and 2021, and again, we're doing a min bucket size here of 7. So you have to have at least 7 data points on the left or right side of any of these splits. The complexity parameter had to increase by 0.05 to fall into this tree. That was what we were doing to prune these models. And both the 2019 and the 2021 models were simply driven by measuring chlorophyll A pigment. And in 2019, that change point was right around 69 micrograms per liter. And in 2021, that change point was right around 62. And so, those numbers, when you're looking at chlorophyll measurements, that's pretty close to each other. And so basically, when you have, let's just call it less than 60, you tend to have micrograms per liter of chlorophyll A pigment, that you tend to have lower microsystem concentrations than when you have greater than 60 micrograms per liter of chlorophyll A pigment in the water. And again, even when Alyssa used all of the data, not just the growing season data like I showed you earlier, you end up with a little bit more complex model, and this model ended up explaining around 74% of the variation, whereas these two models in 2019 and 2021, only explain between 21 and 27% of the variation in microsystem concentrations. And again, the first split here in 2020 with using all data was with temperature. And then there was a little bit of a seasonal split, if the samples were spring or winter. If that answer was yes, you had much lower concentrations than when you had temperatures that were less than 27, but you fell into the fall or summer time frame. And then it split with chlorophyll RFUs. When water temperature was greater than 27 degrees Celsius, you had a split with RFUs. If you had low chlorophyll, you had relatively low microsystem concentrations averaging around 0.4 micrograms per liter. And when you had really, when you had high chlorophyll raw fluorescence, then the data split with the particulate N ratio. And it was when you had an N ratio less than 50 that you tended to have the greatest microsystem concentrations. And so, again, these models are subject to change because it does matter how you prune these and how you set up your minimum bucket size with this type of analysis. But it's very interesting that, at least we thought that each year, 2 years were relatively not too different, but you definitely can't come up with one simple decision tree model like this and necessarily have it apply to all years. And some years can get way more complex than other years. And the next step is to try to figure out and answer that question, why? Why are we seeing more complexity in some years than we do in others? So the next big question that I'll kind of talk about is, what is the connection between cyano-HABs, nutrient availability, and microsystem? In mid-June of 2019, Baylor University ran bioassays on the phytoplankton community at Lake Fayetteville, looking at microsystem, nitrogen, and micronutrient availability. They came out and sampled in mid-June, so that was kind of right after we had this peak in microsystem at our dam site. But it was when nitrate, had decreased down and was pretty low at this site. Nicole Wagner has published this in Harmful Algae. That reference is right here if you're interested in reading the full paper. I'm just going to pull out, with the bioassays that was done with the water from Lake Fayetteville, what did we find in terms of how did we influence microsystem concentration? The first thing I need to point out is that in this experience, in these bioassays, phosphorus limitation was removed. So all of the treatments had a baseline of soluble reactive phosphorus in them around 0.1 milligrams per liter. And then they had differing nitrogen level treatments from low, medium to high, and the high is roughly 1 milligram per liter of nitrate. And what we saw was, high nitrogen resulted in an increased total microsystem production, including specific forms of microsystems such as LA, LR, RR, and YR. And so this was really interesting to us, that at this lake, when you had this algal community and pulled it out of the lake and used it in these bioassays, when they had phosphorus and then you spiked them with nitrogen as well, you ended up increasing the concentrations of microsystem observed in these lakes. And so, with that in mind and kind of knowing that, in 2021, I have a graduate student working on her master's in biological engineering, Lily Haddock, is, we, in collaboration with Thad Scott and Nicole Wagner down at Baylor, is we got set up with an incubator and we were going to run our own nutrient bioassays. And in this time, instead of removing phosphorus limitation across all the treatments, we had 6 treatments of control. So basically just the lake water. And then we had the lake water enriched with low level phosphorus, 0.025 milligrams per liter. We had the lake water enriched with a high level of phosphorus, 0.1 milligrams per liter. And then we had, basically their high nitrogen treatment, 1 milligram per liter of nitrate nitrogen. And then we had a low P in and a high P in. And so those are the 6 treatments that we had. We had 4 replications per treatment. And, we fit all of this in our incubator. And we would incubate these samples at whatever the below surface water temperature was at the time that we collected the water from the lake. And I will note here that, a picture earlier, before we started realizing what the concentrations of microsystem were, as Brad and I were sticking our hands down in the lake to collect the water samples. Now that we know what we're dealing with, we're wearing a little bit, proper PPE, at least on our hands, to limit our exposure to this water now. I'll give you a little bit of the chlorophyll A results really quickly. Just so you kind of know what's going on and what's limiting algal growth. It's probably no surprise to most of you. This is a hypereutrophic reservoir and it stays that way throughout most of the growing season. At times we see really, really high concentrations of chlorophyll A pigment in the water, and it's almost typically greater than roughly 20 micrograms per liter throughout the whole growing season. And you can see here's 20 of the bioassays that Lily had completed, and we kind of have the time frame from a calendar perspective right underneath that, so you can kind of see how they go through time. So if you look at, basically from April through early June, what we saw at Lake Fayetteville was if you had to add phosphorus to these treatments to stimulate any growth. And that comes down to, we know that nitrate is still in the water column at this point in time. And it's sometime in the, in the June to July timeframe that nitrate gets really depleted in this water. And that's exactly what we ended up seeing in this. It's like, as soon as we move into that, pure, summertime growing season, more the July through, almost October timeframe, what we see then is basically we see, the addition of nitrogen alone or even phosphorus alone really doesn't stimulate that much algal growth in terms of measured chlorophyll A pigments in our bioassays. It's when we add the low, when we add phosphorus and nitrogen in combination that we stimulate growth. And the more phosphorus that we add, the greater the growth is stimulated in that. And then, kind of no surprise when the lake starts mixing, and this is, late October or, early to mid-November at this point in time. What we end up seeing here is the lakes mixing. We don't see any response of nitrogen alone, and we kind of see a stair-step response, you know, with low-level phosphorus and then our high-level phosphorus treatment. And you can see here, you know, the first one, bioassay 19, that's when the lake is just starting to mix. So nitrates just starting to be available in the water column. And you can still kind of see a little bit of effect of phosphorus plus nitrogen. But when we move into the last bioassay that I have the data for here, bioassay number 20, you really see that it's, you know, it doesn't matter how, you know, what matters is how much phosphorus you're adding at that point, not nitrogen. And so the next thing we'll do is we'll walk through all these bioassays and what were the microsystem concentrations that we measured in these bottles that we had incubated in the lab. And you can kind of see here, zooming in on Lily's data, and you know, and this is the microsystem concentration in Lake Fayetteville in the water before we started the bioassays. You can kind of see we have that first little peak going up to a high of 1 microgram per liter. The microsystem goes back down as we go into late summer. It stays pretty low, and then it starts creeping back up. And you can see here that secondary fall peak again when we start seeing greater or measurable and greater microsystem concentrations at Lake Fayetteville. The first thing I'm going to do is show you data from these first 6 bioassays. And the reason I'm going to group those together is we basically did not see a change in microsystem concentration between treatments. There was definitely a shift in the magnitude of concentrations that we saw, which reflected the initial concentrations that were in the lake, but the addition of nitrogen nor phosphorus did not stimulate growth or did not stimulate increases in microsystem concentration between these treatments. When we move into bioassay number 7, we start seeing a response where the addition of low-level phosphorus plus nitrogen is different than the control in the nitrogen treatment. And then when we add high phosphorus plus nitrogen, we have even greater microsystem concentrations. And so, you know, this is right when that lake's transitioning to where nitrogen's being depleted from the system. The crazy thing was, is a week, you know, almost a week later when we started the next bioassay, we saw something that was, you know, completely different. When we added any of the treatments that had nitrogen in them, we actually had lower microsystem concentrations than what we saw in the control or what we had. And so you can see here, I mean, the reduction in the microsystem concentrations here, and you know, and so we might actually be promoting growth of a non-toxin producing cyanobacteria in this lake. And this is one of the algal species. I'm not going to try to pronounce that because I butcher it every time, but that's one species that we always see in Lake Fayetteville, and it's not a toxin producer to the best of my knowledge. But then when we move to the next series of bioassays, and this is pretty much what we saw from bioassay 9 through 18. And so that's basically from July through October of 2021, this past summer, is what we observed is very similar to what we saw with the 2019 study that we did with Nicole and Thad down at Baylor. It was the addition of nitrogen that ended up increasing microsystem concentrations in these bioassays. And that was consistent across every single bioassay from 9 through 18. These are the type of results we saw. The magnitude of concentrations varied a little bit in between experiments, you know, were reflective of what the initial concentrations were that in the lake water, but the response was not different across those 9 bioassays that we had then. And then when the lake started mixing, we started seeing a little bit different response with microsystem. And really it became, you know, because now the lake's mixing nitrates, you know, the ammonium that was down in the hypolimnion has been nitrified. There's a little bit of nitrate in the water column, but we still don't have much soluble reactive phosphorus in the lake. we see any addition of phosphorus basically increases microsystem concentration over the control in those last few bioassays. And so, at least to summarize what we're learning specifically from Lake Fayetteville is when nitrate is highly available in the lake water and the cyanoHABs are just forming, microsystem response to nutrients is highly variable, but it's not different than what we're seeing in the control. At that moment when the nitrate supply diminishes, the addition of nutrients, particularly both nitrate and phosphate, stimulate algal growth and increases in microsystem concentration at Lake Fayetteville relative to the control. What's up with that crazy experiment showing nitrogen limitation of microsystem production? Again, we think it's promoting the growth of a species that may not be producing the toxins. But then when nitrate was gone from the lake water and microsystem concentrations were low, nutrients, particularly nitrogen or nitrate supply, increased microsystem concentration in these. And one of the questions really that we have is, are we stimulating microsystem production, or meaning like increasing the amount of microsystem in the algal cells on a cellular basis? Or are we just increasing the growth of algae that have a certain amount of microsystem in them? And, we didn't go into this study thinking that that's what the question we were going to answer. And so we don't have ash-free algal dry mass, to try to put microsystem on a per gram basis of algae that we had in there. So what we're trying to do is we're going to try to use chlorophyll pigment as a proxy to normalize microsystem production. And we're just now looking at this, and we took bioassays 15, 16, and 17, and these are 3 bioassays where we saw a relatively similar magnitude and concentration and similar response with nutrients. And that was, basically you have to add nitrogen to the system to see an increase in microsystem concentrations in the lake water. And if we take that data, you can see the graph right below that, and that's microsystem divided by chlorophyll pigment concentration times 1,000, just to kind of get it on a normalized basis. And in bioassay 15, the addition of nutrients did not necessarily increase microsystem production. It just stimulated the growth of algae that had microsystem in them. But in bioassay 16, the addition of low phosphorus did increase microsystem production relative to the control. And when you look at bioassay 17, the addition of nitrogen stimulated microsystem production. And so, first off, I'll admit, maybe normalizing this by chlorophyll is not the right way, but I do think it's interesting that, the thought of it, we may just be increasing the growth of these algae that have microsystem in it. And at times we may also be increasing cellular, the production of microsystem on a cellular basis. And so, in the future, that's something that's definitely on our minds to try to tackle at this lake. I've got a new addition to my research team, Brittany McIntyre. She's getting her master's in environmental dynamics. The big question is that the city's asking is, how can we manage or treat these cyanobacterial HABs at Lake Fayetteville? You have the traditional copper, products that could be used. Biochar is also being thrown out there. Brittany's actually starting an experiment this week to start look at, a commercially available biochar and how does it, uptake nutrients from reagent grade water. And then we'll move from there to see, how does biochar, influence potentially microsystem production and algal, and algal growth in bioassays like we're doing. And, we're just now on the starting to plan her research out for the next year. Our future plans is we are getting algal IDs for the plankton toes. Basically since like 2020, every time we've been sampling the lake, we take a plankton tow until we kind of build up a certain volume of plankton in the bottom of our tows. So it's going to be a qualitative analysis of the proportion and number of species. We're using Dr. Reed Green, who has a private company, Lake Diagnostics LLC. He's going to be doing the algal IDs for us. We've also, you know, during the summer of 2019 and 2020, we were able to get out and sample the lake, you know, every meter to the bottom across the dam and up the lake, and kind of get at a spatial vertical analysis. And we measured, you know, all these physicochemical parameters in each one of those water samples. And, you know, we're going to look at we're going to, you know, analyze that data in the same way that we're analyzing these data from just those 3 sites on the northern shore of Lake Fayetteville. And as I mentioned earlier, it's, you know, really where we see these the high concentration of microsystem is in that surface scum. And we collect that by just barely putting the tip of a bottle underneath the surface and filling that bottle up. And, you know, one of our next things we need to do is we need to start seeing, can we link the physical, chemical, and algal properties that we're measuring below the surface to the microsystem concentrations and what we're finding in those surface scums. We haven't even looked at that. And but, you know, that's something that's, you know, really on my mind. And, you know, our plan is we're going to continue to study Lake Fayetteville. I mean, it's 10 minutes from my lab, so we couldn't be any more lucky and unfortunate to have a lake like that with the cyano HABs. And so, but we're going to continue studying this lake. We're currently in the planning phases of kind of what we're going to do next. So if you have any thoughts after this symposium's over, please give me an email. I do need to acknowledge my co-authors and collaborators, Brad Austin, Alyssa Ferry, Aaron Grants, Lily Haddock, Abby Lassiter, Brittany McIntyre, Thad Scott, Nicole Wagner. These are all listed in alphabetical order from the Arkansas Water Resources Center and Baylor University. In particular, the nutrient bioassays have been done and conceptualized with Thad Scott and Nicole Wagner from Baylor University. And most importantly, the funding for this research was provided, you know, by the Water Resources Center through the USGS WRRI 104B program, and as well as the USDA NEFA Hatch program and the University of Arkansas System Division of Agriculture. So, you know, without this base funding that a lot of us have access to, and that the Water Center have access to, a lot of this research would not have been able to be accomplished. So with that, I will be glad to allow the Q&A panel to start. Thank you very much for your time today.
Thanks, Brian. Let's see, we will find your top rated question to start with before we move to the rest of the questions for the panelists. So the top rated one for you was, oh, it keeps moving because people are voting. Let's see if I can find it. Okay. It was from Jeffrey Flory at the 1854 Treaty Authority. He asked, could the low nitrogen levels with high cyanobacteria be due to the use of nitrogen to increase the cyanobacteria population?
Yes, I mean, that's absolutely what we see in this lake is when that nitrate gets drawn down in that June timeframe, and it goes from 1 to 2 milligrams per liter down to below detection limits. That's when we start seeing an increase in phycocyanin RFUs. And so it's definitely the drawdown, you know, of the nitrate increases cyanobacteria growth. But one interesting thing that we see is there's kind of a lag effect before we start seeing the toxins. We see the blue-greens come in first, and then it's a little bit later on, typically each year, when the toxins start increasing. So that's a great point.
Okay, and he's got kind of a 3-part question. And since we have time, I think I'll just do the other 2 parts. what was the nitrogen levels prior to the cyanobacteria population explosion?
I did kind of answer that, but I'll reiterate that. This lake, kind of the baseline during winter in this lake is around 1 to 2 milligrams per liter. And so that's typically what we see throughout the winter season. And then that slowly starts declining as that cyanobacteria explosion occurs in this lake.
Okay, all right, great. All right, well, thank you, Brian. So we're now going to move to the general question and answer section. Thank you to all our speakers. We really appreciate your time and expertise. So could I ask that all of our speakers turn on their video if they're able? And then I'd also like to remind everyone to check out the Algal Bloom Action Team's website. And one of our team can drop the website into the chat again as a reminder. So there you'll be able to connect with the team and also subscribe to other events. So we are going to draw our initial questions and comments from the Q&A function on Zoom. So I'll start with the questions that had the highest upvotes. So just a reminder to put your questions in the Q&A box. Please let us know, most importantly, who the question is directed to, or if it is directed to all panelists. All right, let's get going. So the first question I see is from Bob Clement at the EPA. This one is for Hamed, I believe. Toxins bioaccumulate. Can you discuss the state of knowledge on low-level health effects? What is the state of our knowledge? Big question.
Good question. There's no acute effects of low-level cyanotoxins. However, they bioaccumulate, so the chronic effects depends on the cyanotoxins that we're talking about, but they have damaged the liver and reproductive system. I think, if I'm not mistaken, if you contacted any water surfaces during blooms, it's funny, one of my sampling sites was Lake Elsinore, California, and I saw that people, they weren't swimming, but their foot was in the water, and I saw redness, and they said that they feel like burning and tingling in their feet when they stayed there for a long time. So I think that's what I have. Yeah.
Okay, great. Thank you. Next question is for Nafisa. Have you imagined, and this question was asked in a couple different ways, have you imagined how one would scale up collecting the nanoparticles in surface water for reuse?
So basically the plan is to make a separate unit. So we are thinking of like that, the separate unit. And currently we have actually a very long magnetic tube. And the thing is, all the nanoparticles would stick to it. And after that, it would be collected and can be desorbed in sodium hydroxide solution after collecting it. Yeah, I think, yeah, that's how we can collect the nanoparticles.
Another question for you, Nafisa. Have you calculated how much product to use in a lake for treatment, and how much product could be magnetically collected after a treatment?
So I have not calculated this for surface water yet, but our next plan is to, like, from water sample, after interacting with the nanoparticle and doing the magnetic separation, we are planning to calculate how much ferrous and ferric ion is still there after the nanoparticles are collected from the supernatant. So that would actually, that concentration would help us to determine that how much nanoparticles have been magnetically collected after using for treatment.
Okay. Another one for Nafisa, and I'll just add that there does, as was noted in earlier sessions, there's a bias for the earliest presentations, but I promise we have questions for everyone and we will get to them. So Nafisa, would you imagine this technology to be applied in treatment plants or in reservoirs?
So as I've mentioned before, this lake water was from recreational activities, the one I used for collecting water. So our plan is to build a separate unit and add nanoparticles there, and from there add this magnetic tube, and then do in a unit by unit process, not like just directly adding to the reservoir, but a separate unit and treating that, and then collecting nanoparticles from that unit.
This one might be a little bit of a repeat, but I'll just ask it just in case. Nafisa, to your knowledge, has there ever been any in-lake treatment applications?
As far as I know, most of the literature I've gone through was mostly water samples done in lab test, but as far as I know, I have not come by any literature or paper that directly used in surface water yet. Most of them were batch experiments in the lab.
Okay, thank you. The next question is for Hamed and Nafisa. What do you see as the biggest technical and practical challenges in scaling up catalytic treatment technologies for use in either a treatment plant or a water body?
I think one of the challenge could be that how much is still there, the nanoparticles is still there, and how much from surface water, how much actually we are collecting. When we are doing lab test, we are just adding a little bit amount because the vials sample size is less. And that's one of the issue. And another one is that even though from the nanoparticle I have used, it's gamma iron oxide, titanium oxide, even though it's mentioned that titanium oxide is of low toxicity, but however, tests actually need to be done to see that how it's affecting other aquatic system.
Oh, Hamed, looks like you're still muted.
Yeah, sorry. And to add to what Nafisa just said, I think one of the biggest hurdles that we have for using any treatment process in surface waters and in drinking water treatment plants is how we are going to make it efficient. Because when we are doing batch experiments, we can control everything. But when we're scaling up the treatment process, it's going to be more difficult to control all the environmental conditions. We have competing ions, we have other contaminants present. So I think that is, in my opinion, is going to be the most problematic thing that I can think of.
And Hamed, you touched on this a little, but are the reactions sufficiently specific and well understood to ensure that there would not be unintended side effects to water chemistry?
That I don't know yet. I am working on it. I started my work a year ago, so I'm just trying to learn more about what is happening with the cyanotoxins when they are exposed to photocatalysts such as bismuth tungstate. So sorry, I don't have any answer for that.
Okay. Nafisa, any thoughts on how this might, any unintended effects to water chemistry?
Yeah, I think as I mentioned before, that on the composite nanoparticle is titanium oxide. It's considered low toxicity, But however, specific tests need to be done to see that how it can affect like other like aquatic system, other living organisms in the surface water.
Okay. All right, one more for Nafisa. Do you think that there will be a chance to only be able to treat swimmable areas around beaches? To
That
focus it on the areas where
Sure.
people would be swimming, yeah.
Yeah, we have to think further about like only covering only that portion, or how to apply properly in surface water. Yeah, currently I cannot say like specifically like whether it can be applied.
Sure. Okay, Hamed, is there any research about the effects of the iron nanoparticles on metal chemistry in the water, like if the water has other potential human-caused chemical properties? And you might have already covered this.
I believe during my presentation, I talked about iron modification adsorbents. I don't know exactly how iron would like touch the water characteristics and chemicals and chemistry, sorry. But I know there are 3 available articles that talked about iron adsorbents. So that's it, yeah.
Gotcha. Okay. All right. This I think is also for Nafisa. Are these nanoparticles commercially available? And if yes, any idea on how much they cost or how much it would cost to produce them?
The nanoparticles actually were lab synthesized, and I am not sure whether they're commercially available, because I don't think so, because otherwise we would have also tried that one to see how my lab synthesized nanoparticles work compared to the commercial one. And currently I don't have that idea that how much it would cost, because I don't know the like cost of the reagents. Yeah.
Okay. Brian, question for you. Why did you choose to plot microsystem versus particulate nitrogen to phosphorus? And you got a split of 59-1, as opposed to plotting against dissolved, which is bioavailable form most used by this cyanotoxin.
Catherine, thanks. Well, first is actually we put supply nutrient ratio into our CART model as well. And what kicked out in the steps of that decision tree was the particulate N to P ratio. One other problem with using the dissolved or the supply ratio was, quite often most of the nitrate and the soluble reactive phosphorus was much less than our detection limits. And we have pretty good detection limits in our lab. And so it really distorted what those ratios would actually be. And we didn't feel like we had a good measure of what the true supply N to P ratio was. But what I showed you in those plots is what the statistical model kicked out as being most important.
Thank you. I think this is a question for anyone who would like to answer it, but it's sort of a theoretical but a very important question. Can anyone provide, the cost of treatment for cyanotoxins, the cost of it in drinking water, as opposed to doing nothing? So is anyone aware of any sort of good studies or good economic analysis of comparing the cost of some of these treatments versus, the societal cost or the environmental cost of doing nothing? Big question.
That's a great question. I don't know of data that exists. I do know as an engineer from the treatment perspective, if we can keep these toxins in the cell, It's really easy for us to remove them through our filtration processes and different things like that in water treatment. It's when these toxins are, the cells lysed and they're released into the water, it's always more expensive to remove things that are, free in the water versus in algal cells. So that would be the only thing I could add.
Mhm. All right, next one. Let's go to this one for Jin. How many freeze thaw cycles are you doing to get the microsystem results?
I'm sorry that I cannot answer the question because the microsystem measurement is performed by the Iowa DNR. And if you are really need the answer, please email me. I can contact. I can help you contact with the Iowa DNR, or I can ask them for you. And yes.
Okay, yeah, because there's a second part which is also sort of
Yeah,
specific.
I cannot answer
Okay, yeah.
those questions.
It was about what's the lag time between when you collect the samples and when you get the results. But yeah, another question for the Iowa DNR.
Yeah. Yeah, that is true. And actually, my background is the environmental microbiology. So I'm the molecular analysis part was done by myself, but the other data, I just collect all those data after the season. So yeah, just yeah, please email me if you really want to know the answer. I can help you with that.
Another question for you, Jin. Did anyone suggest including data collection for a trophic level evaluation, since you have the capacity to use big data? That may be another parameter to look at.
Yes, I think that's a very thoughtful, good suggestion. What we measured were included, not because of we thought those are only the important factors. Just we try to collect the data what people usually collect for in this type of study. So yes, there could be another important factors we can use, and we can collect those for future future study. But the sad part is that since if we cannot collect, if we don't have the data set for the previously analyzed samples, then those would not be effective for those old samples. So that's the tricky part of the data science, I guess.
Okay, sounds good. Question for Brian. Did you measure the nitrogen and phosphorus retention at Lake Fayetteville? And if so, how do you measure that?
We have not currently been measuring that. Erin Grants did research on that, and she published a paper on Lake Fayetteville and limnology and oceanography. And basically, we gauge the inputs and the outputs to the lake, and looked at how much nitrogen and phosphorus is flowing in versus flowing out. And quite honestly, that study was also comprehensive, and we looked at atmospheric deposition as well as calculated nitrification rates and denitrification rates. And so kind of in my study side description, about 90% of the phosphorus that comes into Lake Fayetteville is retained within that small reservoir, and about half the nitrogen that comes in is lost through the output, flows out of the reservoir, and that other half that's stays within the reservoir is actually lost through denitrification.
Okay, sounds good. Yeah, but I can see some chatter in the chat function. Sorry if I'm not doing a good job with your questions. We're trying to sort of manage all the questions and get to the ones that are first. And so I'm not able to always keep track of the answers. So sorry for that, but we'll try to get everything answered. And I think we have everyone's contact information added. So if you have had a great question that I totally butchered, you can please feel free to contact some of our panelists and I'm sure they'd be happy to answer it. Okay. Next question for Jin. You said some lakes have issues and some have no issues with cyanoHABs. Does Iowa have a standardized sampling protocol and quality assurance program plan?
Yes. So the Iowa DNR has very well documented protocols, and previously I answered one of the questions about which point the sample was taken. So I looked it up. I had one of the documents. So they have all the procedures in order and in figures. So they train they hire 3 or 4 people every year during the season, and they train them and like they sample in 9 different points in one lake. So based on how deep it is, and they mix them all together for analysis. So yes, they have a very well designed protocols and plans.
Okay, thank you. So going back to a question for Nafisa, this is a bit of a long one. If I understand the results, you get less than 30% reduction in cell counts, assuming the cells you counted were still alive and not leaking material that could diffuse to the surface where ROS is generated. But you get 99.999% reduction in the genes that code for toxin production. So the question is, if I am interpreting this correctly, the big if is really, it sort of suggests that the photocatalyst is either selectively destroying the MCYE containing cells, or it is influencing the cells to stop encoding and maybe recycle the MCYE material if already produced. Did you follow that? I know it was a lot.
Let me see what's the question. Yeah, I think, yeah, the cell counts and the Qpcr results are a bit different because the cell counts is actually not like it was done as an estimate, because even if the nanoparticles have like under the optical microscope, even if the nanoparticles are actually like, as it said, that is targeting those MCYE genes, and even if that cell is retaining its shape, it may not be that apparent from the microscope that I used in the lab. So it could be that it's targeting that specific genes and yeah, stop that inactivating that part. Yeah.
Okay, then we have sort of a question and a comment for Brian. Maybe you could look at the depth, shallow lake, wind exposed, phosphorus resuspension, and solar intensity. Any thought about looking at sediment phosphorus concentrations?
Yes, in 2020, we collected sediment cores, and there is a ton of phosphorus that leaks out of the sediments in this lake. And over the 2021 growing season, we actually had a platform that was taking temperature profiles on, on a very short time basis. Unfortunately, something happened in our recharger where It only worked at night because of the way the solar panel was interacting with the batteries, and we could never figure out what was going on. But we do have at least thermal profiles of the lake every evening, all throughout 2021, where we can look at mixing events like the question is talking about. We haven't done that yet, but that was our intent for having that platform out there, is do when mixing events in a small, relatively shallow lake like this, and train some of those nutrients that are coming up in the hypolimnion and get those back into the into the photic zone where these algae are growing. So that's a great question, and that's on our agenda to try to answer with some data we've recently collected.
Okay. Next for Jin, what water quality parameters were collected and analyzed?
Oh, yeah, I'm sorry. I forgot to mention those in the presentation. So the DNR measured microsystems, pH, dissolved oxygen, turbidity, and at our end, another collaborator measured DOC, TKP, ortho-P, TKN, and nitrate and nitrite, and chloride. That's what we measured.
Good memory. Okay, one more for you, Jin. To what do you attribute the alkaline pH readings?
Does it mean the pH of our samples were quite high?
I believe so, but I'm not sure.
Yeah. We don't know why actually, but so the Iowa DNR measured pH on site, and we also measured pH in our when we got the samples here, and those numbers are quite matched to each other. So we just I could just say it is what it is. So yeah.
All right. Another one for Brian. Could you use the P to N ratios instead of just nitrogen?
Yeah, we actually used the N to P ratio as well as the magnitude of each of the individual concentrations in our relationship. And so what you see in those decision tree or regression tree models is whatever factor was most important in each one of those steps, that's the one that popped out. But Microsystem showed a threshold with both the individual concentrations of nitrogen species, dissolved and total, and phosphorus, as well as with the N to P ratios as well.
Another question for Jin. This is an interesting one. Is it possible to imagine this process applied to other Iowa waterways?
Yes, why not? So I mentioned about the 4 key elements. So which was algorithm. We have the algorithm already. So the other 3 is quantity of the data, quality of the data, and the right proper question. So if those elements are there, it can be applied to any type of environment, not only just the water. It can be applied to soil and other systems as well.
Cool. For Brian, what approximate chlorophyll A level in micrograms per liter does the raw fluorescence correspond to?
Yeah, I actually just dug that spreadsheet up to look at it. So the first thing is the phycocyanin raw fluorescence has a much stronger relationship with measured chlorophyll A pigments than does the chlorophyll A raw fluorescence. So when you're looking at those on an individual basis, there's a lot of scatter in the data, but a chlorophyll raw fluorescence of around 4,000 that say corresponds to between 20 and 40 milligrams per liter or micrograms per liter of chlorophyll A pigment in the sample. So that would be the range that you'd be looking at right there.
Another question for Brian. It sounded like you were just starting the biochar study, but they are curious, does the biochar have any metals or toxic issues like PAHs that may be of concern to the lake or aquatic systems?
That is, I saw that question, and that's a great question because, yeah, we're just starting this. I have no idea about the polyaromatic hydrocarbons that, you know, and toxic residues or metals. So that's definitely something we should measure. Is when my students doing this, I'll mention to Brittany, is we should at least do a water extraction and see, you know, at least in terms of metals, what, if anything, would leach out from these biochars. It's a commercially available product, and so they don't really give a whole lot because of proprietary reasons as what the source of the biochar is. But that's a great question and something that I'll definitely have Brittany try to tackle or at least have an eye on.
And Jin, it looks like maybe one of your colleagues at the DNR might have answered one of the questions, 3 freeze-thaw cycles. I'm not sure if Daniel Kendall is one of your colleagues there.
Yes, thank you, Dan.
All right. Next, for Brian, I could see different, I could see different units for chlorophyll RFU and microsystem micrograms per liter in the graphs. Could you explain the relationship between those units?
Sammy, follow up with me via email if I don't get this question right. The raw fluorescence for the chlorophyll is kind of giving us an idea of how much algae is in the water. And obviously the microsystem, that's what we are, you know, that's the toxin that we're measuring with our ELISA kits. If what you're referring to is we actually had different thresholds, and those different thresholds just relate to how the data split before we got to that threshold. And that's really the only answer I can give you based on how I'm interpreting your question.
Okay, and I think this one will be our last question. It's for Jin, and it's a little philosophical again, but Scott asks, if many of us try to use machine learning, will we need to look at increased data storage and processing capacity? Is that a consideration of yours, or, you know, if we were to develop this model beyond where it is today, are there some data limitations that might affect its accessibility?
It can be like if the data set includes like images, a lot of images, or larger volume of data set, then maybe it would need more computing power. But like in the form of the tables, like usually the data set we have only the one file with lots of numbers. In that form, it would not need that much computing power. So I myself use my just laptop for that. And even if a little more computing power and storage is needed, the university's high performance computer just will handle that fine.
Okay. All right. Well, thank you. Thank you to all of our panelists. We really appreciate your time and expertise. So thank you for being a part of the symposium. Now I'm going to turn it over to Rebecca Power for some closing thoughts. And Rebecca is the director of the North Central Region Water Network.
Hello, everyone. Can you hear me? All right.
Yes, I can.
Okay, great. Thank you. All right. Yes. Good afternoon, everyone. Rebecca Power, University of Wisconsin-Madison Division of Extension and the North Central Region Water Network. Thank you so much for being with us today. I learned a ton about HAB monitoring and cyanotoxin monitoring and detection and treatment, and I hope you did too. We thank you so much to all of our speakers for your excellent presentations and for all the Q&A time. We definitely kept more than on time, so we had a good amount of time for questions. And thank you to those of you that are participating with us and all the expertise you're bringing. That dialogue that we're having in the chat and as part of the panel is extremely helpful. Land-grant universities and extension, and this is what we try to do, is connect all of you with the latest research, and hopefully we were able to deliver on that mission today. Thank you so much to our sponsors listed there on the slide for making this event possible. And I also want to give a shout out to our planning team. When things go right, a virtual event can look effortless, and as I'm sure many of you know, it is not effortless. So thanks to our extension colleagues and our Water Resource Research Institute colleagues for all their behind the scenes work. The recording of this session and the PowerPoints that our presenters are able to release will be available in our Harmful Algal Bloom Action, or Algal Bloom Action Team website, which we've put in the chat a couple times, and we'll put in there one more time. For tomorrow, we hope you can join us again. We will begin at 8:30 a.m. Central, 9:30 a.m. Eastern. And we will be talking about forecasting and modeling HABs, certainly important. And we touched on some of those efforts today, but we'll continue to talk about those so we can do our best to be as proactive as possible in mitigating the negative impacts of harmful algal blooms. We will also be covering some emerging research and technologies for HAB detection. So things that are just early on in the stages of development. So we hope you'll join us for the latest. And also, I know some of you commented in the chat, perhaps you were thinking to join for a particular presentation today. And for one of our presentations that was shorter, we didn't have questions to start out right away, so we worked right into the next presentation. For tomorrow, what we'll do is we'll ask you to contribute your questions when you have them right away. So if we do end up having a shorter presentation, we can jump right into those questions, and then if we'll take a pause after that presentation and questions are done, so we can stay a little bit more close to the published agenda for those of you that might be interested in joining for different presentations. So that said, thank you again so much, and we hope that we will see you again tomorrow.